So hello everyone. This is one more seminar from the Statistics Department at Federal University of Minas Gerais. This is our last invited of the semester. Next week we have a seminar from Adriana of her requests from her PhD qualification exam. She needs to do some presentation. So next week is Adriana. But this week, we end up, I think, with a very interesting topic. We'll see very applied, difficult statistical problem that we'll see in ecology. So I have the pleasure that Mike accepted my invitation. If I need to do a small presentation of Mike, it will take too long. So I would just say basic things. I met Mike when I was in my PhD at the University of Connecticut. He well, he is the coordinator director of a center that has some support in the summer for students, and I applied and I got a scholarship there in the summer. That's when I started working with him, and since then we are in touch because he always brings this interesting, complicated, not trivial statistical questions to us. So if you look on his CV, I said a small bio. He has plenty of, you know, students that he advised. He has collected a lot of money for grants. He's the center of, institute. He's a distinguished professor and so on and so forth. So it's a pleasure for me that he's dedicating his time to talk to all of you. And I'm very happy that he's, closing up our seminar this week. Mike, thank you for accepting my invitation, and it's my great pleasure to have you here. And the floor is all yours, and you have around 15 minutes, and then we go for the questions.
Great. Great. Thank you, Marcos, for the kind introduction. It's obviously a pleasure to talk with you and the faculty and students in your department. And as you know, the work that I'm going to present today is very much research in progress that'll hopefully present some opportunities for statistical research on the part of your colleagues, and also collaborations with us here at the University of Connecticut.
Is it not going?
Yeah, I'm trying to, yes, it's not moving. Okay, there we go.
Okay.
So I'll go back. So the presentation today is organized into 5 parts. I'll give you a very brief introduction to the context or motivation for doing the research. Then I'll talk equally briefly about circular statistics, but some of the problems with using circular statistics that involve experimental designs or data sets that have marginal totals. And then talk a little bit about a bootstrap approach that we've taken to try to sort of ameliorate or improve the circumstances. And then try to leave time at the end for comments and critiques from those of you in the office. And although Marcos kindly introduced me as the presenter today, I want to point out that this research is really a collaboration between assistant research professor in the department, Julissa Rosas Sandoval, as well as a postdoc in my lab, Steve Presley. So the context and introduction are relatively straightforward. Timing is everything. From an evolutionary perspective, natural selection should favor behavior of individual organisms that enhance their reproductive success and reduce the likelihood of death. So questions such as when should an organism reproduce? When should it be active? When should it be foraging? These are all important questions that relate to temporal ecology. And the timing of these events are particularly important when there's variation over time in conditions or resources that would result in beneficial activities occurring during only part of the year. This is particularly true for bats, morcegos, my study organism, where the cost of reproduction in these organisms is very high in terms of pregnancy and lactation, in part because they're warm-blooded, in part because they're volant or fly, and in part because they bear young that can eventually reach a mass that's about half the size of their mothers. So deciding when you're going to reproduce and when you're going to lactate are really, really important in terms of the energetic constraints that allow you to survive and persist in an environment. So historically, 3 different kinds of reproductive phenologies have been identified. That's interesting. There's actually 3 diagrams in this slide that you can't see. As statisticians, you'll understand uniform is simply when there's no trend in peaks and troughs in the abundance of activities. Unimodal is when there's a single peak in reproductive activity, and bimodal would occur when there's actually 2 peaks in activity over time. So the cartoons that you couldn't see were hyper simplified versions of what happened in reality. So this is a slide that's an actual phenology of the reproductive behavior of Artivius litoratus, a very common bat in the Amazon of Peru, which is where these data come from, but also in the Amazon of Brazil. And the data come from 5 years, and so they're the cumulative data over a 5-year period. The colored circles represent the proportion of females that were pregnant in the population. The open white circles represent the proportion of females that were lactating in the population. The solid black triangles represent the proportion of males that were reproductively active. The gray shading in the background represents precipitation, and the numbers across the bottom that are in parentheses represent the sample sizes of female bats that were caught in a particular month, whereas the numbers that aren't in parentheses represent the number of males that were captured in a particular month. So most scientific research, at best, up to about 10 years ago, would present a diagram like the one that I talked about just a few moments ago and say, well, that was an example of bimodal reproduction or unimodal reproduction or aseasonality. And in a recent paper, some students of mine and I published came up with rules of thumb to help document when we look at the variation over a year's period, when we can identify a particular peak or mode in reproductive activity versus when we can identify secondary peaks. And these rules of thumb simply help guide decision-making so that you aren't biased to come to one conclusion or another. They're arbitrary but reasonable decisions. And like I say, up until about 10 years ago, that's the best we had in terms of characterizing the reproductive phenology of bats. So using those 2 rules of thumb about identifying primary or secondary peaks, we can apply them to these data, the ones that I just showed you for Artivius litoratus, and it would conclude that there are actually 2 peaks. The primary peak, the one with the highest reproductive activity here in December, with a secondary peak in August, and each of these peaks is separated from each other by a trough or by a great reduction in reproductive activity that allows you to pretty distinctly see that this species is exhibiting a bimodal pattern in the Peruvian Amazon. And so we published this paper in 2023, and it became the motivation for all the rest of the material that I'm going to talk about today. When we submitted the manuscript for publication, we received comments back from one of the reviewers, or actually, that we should essentially do circular statistics on the data so that we could evaluate whether or not the patterns we saw deviated from uniformity or not. And we were sort of excited about this to begin with. And in fact, we applied contemporary circular statistical techniques to all the data we had about bats, but quickly came to realize that although those statistics would come to some conclusions, they were incredibly biased with respect to the data that we were working on. And we presented what were ultimately strong arguments to the journal that the application of contemporary or classical circular statistical techniques would not be appropriate for the kind of data we had. And I'll explain those kinds of data to you shortly. But that pattern and the critiques from the reviewers and our subsequent responses to those critiques became the motivation for, at one point, talking to Marcos about what he thought about our position on this, but also for us exploring a second paper that we're currently working on to talk about ways to overcome the limitations of contemporary circular statistics when, in fact, you have marginal totals. So now what I want to move on to is a brief overview of circular statistics, why they exist, and how they could be applied to data such as that on reproductive phenology of bats. So one of the easiest ways to think about circular statistic is when the response variables aren't following a linear pathway, that they're actually interconnected or cyclical, such as the degrees that represent distances, or the hours in a day when you're talking about diel patterns, or the months in a year when you're talking about annual patterns. So this is an example of when we look at the circular distribution of degrees, which is essentially polar coordinates, nothing fancy about that. You can see that the distribution of observations is relatively uniform across all the possible angles or degrees of direction that would be available. So the classical approach would be you collect 100 different birds, you release each of those birds from a central locality, you record the direction that the bird flies in, you mark that directional point with a red dot, you do that 100 times, then you get a distribution of dots like this that would suggest that, this species has a uniform dispersal from the point of where I released it. As opposed to an alternative hypothesis is that the birds have some sort of homing device where they are able to identify where they have been transplanted to, and they would fly back to their original location. And that would indicate sort of a non-uniform response, where although there's birds that move in every direction, the preponderance, a large number of birds, moved in this northerly direction rather than any other direction. So circular statistics are designed to evaluate the distinction between uniform and non-uniform responses when the data appear in most accurately in a circular format. And this is critically important because arithmetic estimation in these cases will not estimate central tendency or dispersion. And a simple example illustrates that. If I had 2 points, one at 350 degrees and one at 10 degrees, the mean of those two measurements would be 180 degrees, the exact opposite direction in which most of the bats were moving. So one was moving at 350, one was moving at 10. The average of these two would say 180. They weren't moving toward the south, they were moving to the north. And so the adjacency of 0 and 350 create problems. And so circular statistics are designed to take into account that fact by essentially transforming these in values into measurements that are consistent with polar coordinates, and then looking at the data in that fashion. And at least in the ecological literature, the two tests that are most often utilized for evaluating circular distributions and distinguishing between uniform and non-uniform distributions are the Raleigh and Hermans-Rassens test. Each of them are pretty popular. They differ, however, in terms of the alternate hypothesis that they evaluate. The Raleigh test has a directional alternative hypothesis that the preponderance of individuals are located along one directionality in the circle, whereas the Herman-Rassen test only addresses issues of non-uniformity. And recent papers by Landler et al. Have suggested that Herman-Rassen should be the test of choice if you only were going to pick one, because in fact, when the alternative distributions are multimodality, it's the preferred test over the Raleigh test. But even if the alternative is unimodality, the Hermans-Rassens test performs essentially just as well as the Raleigh test. Nonetheless, we've used both of them in this research to try to see their relative sensitivity to the issues that we'll talk about. So we did this. We took data like those for Artebius literatus that we, by using rules of thumbs for identifying distributions, and where we classified this species as bimodal, we applied the Raleigh test and the Hermans-Rassens test, and each of them said, yep, you were right. Your assessment of the pattern was that it was not uniform. In fact, it was modal, or at least it was non-uniform in the case of the Raleigh test. And in fact, these distributions that are classically shown by biologists in these kind of diagrams that are linear in expression are probably most appropriately illustrated by these kinds of Rose diagrams where the data are displayed in the same radial fashion as is the data itself. And you can see that most individuals reproduced between July and December's, with hardly any reproduction in other months. This is true based on absolute numbers, and this is true based upon percents. And an important point to both the Raleigh test and the Hermann-Rassen test is based on the number of individuals that were associated with a particular time interval, not the percentage of individuals. And this becomes important when there are marginal totals. And so what I'm going to introduce now is this sort of concept of what is a marginal total in the context of reproductive phenologies, to expose the problem for analysis, and then also to motivate what we hope is a reasonable solution to that problem. So circular statistics, like the Raleigh test and the Hermann-Rassen test, have a variety of assumptions. Each individual essentially has the freedom to choose a time interval to be active. And by active, that's the response variable. In the case that I'll be talking about today, it's just going to be the proportion of pregnant females. Moreover, the number of potential individuals per time interval is not fixed by any constraint, sample size or sampling success, and it's also not multinomial. Remember back in that little example where I showed the compass, each individual was released and it chose a direction in which to move. So there was no dichotomous choices. The number that would move at any time interval wasn't limited by anything except the total sample size that I had. And then finally, the assumption is that the number of individuals per time interval defines the pattern that we're interested in exploring, rather than the proportion of individuals per time interval. And these three assumptions, we think, are particularly inappropriate in their application to data on reproductive phenology of bats collected by a design that I'll talk about shortly. Again, this is unfortunate that the actual figures aren't showing up on the slide. But so in order to test the, actually, Marcus, might it be possible for you to share the slides at your end, and maybe we're not going to have that problem? I'm going to see if...
Yeah, let me try that.
I'm going to see if the others... now they're showing up. Okay.
I can share if you feel like, or you can move on. What you prefer.
Well, I'll try to describe that. I don't think it's all that hard. So, okay. So we created a 2x2 matrix to guide our construction of exemplar data sets. So one is based on the idea that we will construct the data so that the percents are either uniform or non-uniform, and that'll also be reflected in whether the numbers are uniform or non-uniform. And that'll result in a 2x2 classification where we can think of percents as the real patterns and numbers as the data that are used in circular statistics. This will be clear in the next slide, so I won't go into great detail about it here. So this is another visualization of the exemplar data sets that we created. So in the uniform pattern, this is an example where if you look at the number of pregnant females in the populations, it varies a little bit between, I think, 20 and 30, but it's relatively uniform. And that's reflected in a relatively uniform distribution of percentages. Of course, that's true because in this example, we selected the marginal totals to be constant at 100. And when you look at these windrows diagrams, you see that the pattern that you see based on percents and the patterns you see based on numbers are very similar to each other. So in this particular example, the data based on percentages is very similar to the data based on absolute numbers. And the statistical conclusion you arrive at based upon numbers corresponds to the pattern that we constructed for the data based upon percents. So in this particular case, both the Raleigh test and the Hermann-Rassen test rightly identified the data as being uniform. Alternatively, we constructed an exemplar, where there was big variation in the number of pregnant individuals per month. In most months, it was 20. In June and July, it was 100. And this is also reflected in the variation in percent pregnant in the population. And again, you look at the windrows diagrams based on numbers and percents, you get exactly the same pattern. That's because the marginal totals are constant in all 12 months. So these patterns look very much alike. And in fact, the Raleigh test and the Herman-Rassen test come to the conclusion that we would have expected. The proportion of females in the population is highly modal, with peaks in pregnancy in July and June. Whereas on the previous example, there was no modality. There was sort of stochastic variation in the percentage of individuals that were pregnant in the population. So these were two examples that we constructed where we thought the two circular statistical approaches would come up with the right conclusion, and they did. But remember, we also constructed examples that represented mismatches, circumstances where the pattern based upon numbers didn't look like a pattern based upon percents. So notice here we constructed the data in the orangish color so that the percentage of females that were pregnant in the population was constant, but the absolute number of individuals in each month that were pregnant was highly modal, with, again, peaks in June and July. You can see that in the rows diagrams. The pattern based on numbers says, oh, that looks really modal, but the patterns based on percents is that they were uniform. They were always 50% of the adult females were pregnant. In this particular case, the circular statistical approaches said the pattern is highly non-uniform, but in fact, based upon percentages, it's uniform. We consider that to be a mismatch. Similarly, in the blue example, we constructed the data so that based on percents, there's a peak in reproductive activity in June and July, where it's twice as active as it is in all the other months. But because we varied marginal totals, we were able to construct an example where the number of pregnant females was constant. If you applied the classical statistical approach, unsurprisingly, it says, yep, absolute numbers are uniform, and they are. But this is a misleading representation of the proportion of individuals in every month that are pregnant. And the patterns that you're seeing for absolute numbers is not only a reflection of the proportion of females that are pregnant, but also the number of females that you sampled during a particular month. So the fact that we can't control the number of individuals in a particular month that are available to be pregnant or non-pregnant is reflected strongly in the results. Was that you, Marcos?
Yeah, that was me, because if you want me to show something, I would just put in the screen behind. So you'll
Yeah, I think we should probably go to yours, because when I accidentally moved forward, there was another slide there that was outrageously incomplete. So maybe we
that is what I noticed. So can I
yes, why don't you go? You can present the slides now.
There you go. So just let me know when to move, okay?
Okay, sure. So why don't you go to the next slide, Marcos? So what you saw from the previous two slides was we constructed exemplar data sets that, as we expected, the circular statistical approaches appropriately characterized the data. But we also constructed two examples where we thought that circular statistics would come to an erroneous conclusion, and the circular statistics did come to an erroneous conclusion. In one case, saying the distribution was uniform, when in fact it was highly modal based upon the proportion of individuals that were pregnant. And in the other case, saying it was modal, when in fact it was continuous, uniform, with all of the females exhibiting exactly the same proportional reproductive activity in a particular month. So that was the problem that we confronted. And to address that problem, we developed the bootstrap simulation approach. And this slide is a little complicated, but hopefully not overly complicated. So I tried to illustrate the problem and the solution as follows. In the first three columns, we're showing the empirical data. We're showing a number of sampling months that are color-coded: January in orange, February in blue, June in green, and December in yellow. Of course, in real data, we'd have 12 months, but this illustrates the process. In each of those months, we know the number of pregnant, non-pregnant, and total individuals. And I've illustrated those are characterized here and reported here. If we conducted circular statistical, pardon me?
We cannot see your mouse, so when you say here, I don't know.
Oh, I'm sorry. Yeah, okay. So if you move to the bottom of the third column, you can see that we were able to calculate a test statistic or a significance value for those empirical data. So based on these data, you would have concluded that there's no significance, that it's a uniform distribution based upon the data that were available. Of course, you're only seeing, remember, 4 of the months, not 12 of the months. This is just to illustrate the process. Well, remember, from our perspective, the problem arose because marginal totals were fixed as a consequence of sampling success. And so in those empirical months, in January, we captured 200 individuals, February 200, June only 100, December 200. So the number of pregnant females is a consequence of not only the sort of the parametric proportion of females in the population, but also the number of females we just happened to successfully capture in those months. So in order to control for variation in monthly sample sizes, to remove that effect from our statistical conclusion, we did this bootstrap approach where we will do 10,000 iterations. And in doing so, we have to establish a common sample size that we call N sub C. This standardized sample size, well, there's no right answer to what that sample size is. It could be the average sample size that we observed in a month. It could be the minimum sample size we observed in a particular month. There's any other value. For this process, we used the minimum number of individuals that were sampled in any month as our common sample size. So in the 4 months, so you can't see that, in the 4 months that I've selected, June had the lowest total sample size of 100. And you can see that in all the iterations, there's always 100 individuals in all the monthly samples, regardless of iteration. And the next step is to say, well, given now that we're having 100 individuals in each of those months, let's randomly sample exactly N sub C, 100 individuals from the monthly pool with replacement. So for January, for example, we sampled 100 individuals from a pool that had 100 reproductive and 100 non-reproductive individuals. And we obtained 47 and 43 individuals. In February, we repeated that same process and randomly sampled from the species pool, the reproductive pool in February, and essentially 50 randomly selected from a pool of 100 individuals and got 43 and 57. In the month where we only captured 100 individuals, we randomly sampled from that pool, and notice that the only outcome that's possible is 100 individuals that are pregnant and none that are non-pregnant, because the sample sizes are constrained and the pool of individuals from which we select have no individuals that are pregnant. And we did this for all 12 months. During the first iteration, we calculated the test statistic and the p-value associated with that, artificially constructed data. So remember, these data now are constrained by the sample size of 100, but we use the empirical distribution of pregnant, non-pregnant females in a particular month to populate the simulation. And based on that, then we obtained a test statistic and a p-value, and we repeat that steps 3 and 4, randomly sampling from the monthly species reproductive pool, estimating a test statistic and p-value, and we end up then with 10,000 test statistics, 10,000 p-values, each of which was constructed by chance, but constrained by the empirical marginal total for the month in which the data were collected. Next slide, please, Marcus. And so the outcomes could be summarized like this, and these are 2 cartoons. These are obviously not real data, but they show 2 extreme possibilities. In the top example, there's a distribution of 10,000 p-values that looks pretty modal in distribution, and we illustrated that only a small number of those were significant, less than 5%, an arbitrary alpha value, but one that's pretty regularly used, at least in biology. The median p-value was 0.49, the mean p-value 0.47. the proportion of all the simulations that were significant was only 4%. 96% of those 10,000 simulated values was not significant. The equivalent of a 95% empirical confidence interval for those data, or that the p-values fell between 0.13 and 0.95. No matter what criterion you would use, you'd probably pretty convincingly be able to argue that the preponderance of evidence is that these data are not significant. They do not suggest deviations from uniformity. Alternatively, you could have an empirical result that looks like the bottom half of this slide. Or notice in this particular case, the vast majority of the simulated results had p-values less than 0.5, and only in a few cases were p-values greater than 0.05. And in fact, in this hypothetical data set at the bottom, the mean p-value was 0.03, the median p-value is 0.02. 92% of the 10,000 simulations returned the result of significant, only 8% were non-significant, and that the p-values essentially varied between 0.001 and 0.043. Again, the preponderance of evidence from this analysis would have suggested that there were deviations from uniformity. And so in all the rest of the talk, I'll illustrate the results based upon the mean p-value and then come back to the concept of preponderance of evidence. But in preview, the preponderance of evidence is going to, in these examples, corresponds exactly to what the mean p-value told you. So it wouldn't matter what criterion you used for making a decision. Next slide, please. So we're going to move on to those 4 exemplar data sets, the 2 that represented matches and the 2 that represented mismatches. Next slide, Marcus. And what you can see here is that you can see that in the 2 cases of the 2 matches, the rows diagrams for percents and numbers are identical, and the bootstrap results are shown in the far right of the little rectangle on the right-hand side of each diagram. Notice in the purple example, the p-values or the median p-values are non-significant, suggesting a uniform distribution. And for the green example, the bootstrap methods show p-values of 0.001, highly significant, non-uniform values. Again, so in this case, the modification based upon the bootstrap doesn't change the statistical conclusion. The statistical conclusion corresponds to what we had hoped would be illustrated with the exemplar data sets. Next slide, please, Marcos. Now, these were the more interesting simulations because they were constructed so that there was a mismatch between the pattern that you would see based on absolute numbers and the actual pattern that we were hoping to detect based upon proportional representation of the population. And notice in the orange example at the top, the proportional distribution of pregnancy was uniform across space based on percents. And in below, you can see that based on percents, the distribution was highly modal. So again, in the top, the bootstrap approach said non-significant based upon mean p-value. And the bootstrap approach in the blue example says highly significant. So the bootstrap is able to correctly interpret the pattern in terms of percents to correspond to the rightmost rows diagram that we had when we showed those examples. So we felt pretty good that the bootstrap was, at least in the terms of these 4 examples, was not plagued by the same problems as the more pure circular statistics were in terms of being only sensitive to the number of individuals that were pregnant in the particular category. Next slide, please. So in terms of the matches, the bootstrap was successful. In terms of the mismatches, the bootstrap was successful. Next slide, Marcos. So now we want to apply this bootstrap approach to empirical data, not fake data, not cartoon data, not data that were designed to illustrate particular problems with marginal totals. Next slide. And the format for the 2 slides are similar to the ones that you've seen before. On the leftmost diagrams, you see the way biologists illustrate phenologies. And in blue for each of those empirical phenologies, I tell you what in the previous analyses that we derived as the phenology. So Artebius literatus was bimodal. The middle species, Artebius obscurus, was unimodal, and the bottom species, Artebius planirostris, was unimodal. If you look over, if the two rows diagrams for each species are pretty similar, and in this case, both circular statistics and the bootstrap correction of circular statistics came to the same conclusion. The data are not uniform. They're highly non-uniform, indicated by p-values in all these cases of less than 0.001. Next slide, please. So again, this is 3 additional species of bat. The conclusions that we arrived using rules of thumb was that each of them had a bimodal reproductive phenology. And when the 2 rows diagrams on the right for each species illustrate the similarity in the patterns based upon numbers and percents. And in these 3 examples as well, circular statistics, the classical approach, rightly concluded that the data were not uniform, and the application of the bootstrap approach did not change that conclusion. Essentially, the data were again identified as being non-uniform or modal in distribution. Next slide, Marcos. So the conclusions for the empirical data sets, of course, with empirical data, you don't know the right answer, right? The right answer is out there, and we're trying to detect it. But the bootstrap approach was successful, at least in detecting non-uniform reproductive phenologies for data that we previously had said were unimodal or bimodal, with the peaks occurring in tandem, and bimodal with the peaks being distinct and separated by periods of inactivity. Next slide, Marcos. So in this slide, we're focusing only on the Hermans-Rassens test and illustrating the concordance of interpretation based upon a number of metrics that we could have used. So in yellow is the result when we chose to use the average N, the average monthly sample size, as the basis for tests. And in the right, we chose that common sample size to be the minimum N. So essentially, in orange, you should have lower power than you do in the yellow, right? Because the sample sizes are different. So if we just look at the results for, again, Hermans-Rassens test, you can see that in the case of, we'll pick one that's interesting here. Well, all of them actually. If you look at uniform, all of the data in the top yellow row says things are uniform. There's no deviations from uniformity. And that's true, as Marcos is illustrating, for the yellow data where we used average N, or if you go to the orangish column, again, same pattern, non-uniform. And in fact, if you look down all of these data sets, whether it's for the 4 exemplars or for the 6 species of bats, they're highly concordant results. That no matter what approach we take in looking at whether or not a response from the bootstrapping was significant, suggests that things are either conclusively modal or inconclusive or conclusively not modal. There's one slight exception to that, and that's the top species, Artebius literatus, in the right when we have the smallest sample size. Although a lot of information, many of the metrics, the median p-value, the mean p-value, the lower bound of the confidence interval, all suggested significance. The upper bound was in the range of non-significance, maybe marginally significant, but not significant. So we're not exactly sure how to adjudicate or how to decide in cases where everything isn't in accord, whether or not we have evidence for significant deviations from uniformity. And so that's why we think the concept of the preponderance of evidence becomes important. And at some levels, it's not as satisfying when you just don't have a p-value and you can say it's significance or not, or at least that's what a lot of biological reviewers like to see, that the decision of significance is taken away from the researcher. But in reality, the preponderance of evidence is sometimes a stronger approach for determining whether or not patterns are real or simply artifacts of sampling. Next slide, please, Marcus. So in the last section, I've tried to leave about 20 minutes in my presentation to engage you all, hopefully, and with respect to comments and critiques about the work that we presented here. Next slide, Marcos. And in particular, these are some of the questions that, well, we'd like you to at least think about either now or later. In that, do you think the bootstrap approach is valid? What do you think about the use of minimum monthly sample size versus average sample size or some other standard? What do you think about using mean p-values or median p-values or the confidence intervals as the p-values or the criterion of the preponderance of evidence and making conclusions about whether patterns are real or not? Can you think of methodological modifications that would improve the efficacy of our approach? Do you know of alternative approaches that might be more appropriate than the one that we used? And finally, remember in all of these analyses, what we're really distinguishing is uniform from non-uniform or directional. We're never able to, in these analyses, statistically evaluate whether or not the patterns are unimodal or bimodal. And that would be important for us to be able to do in the future. So next slide, Marcos. So I want to acknowledge that the analytical phase of this research was supported by 2 grants from the National Science Foundation in the US, and that the data gathering phases were similarly supported by 2 different NSF NIH grants to work on the ecology of infectious disease on bats in the Peruvian Amazon. If you can go back to the previous slide, Marcos. And so I want to end with this and open up for any questions, comments, critiques, whatever. And thanks so much for the opportunity to chat with you.
So, Mike, obrigado. So we have some, as I said, some small delay so people can ask questions. And meanwhile, I'll have some comments to you so you can think after we talk. First, I'm not sure that you can take means of p-values and use it as a kind of p-value metric, because the p-value itself is a probability, and I'm not sure if the distribution of probability measures makes sense. So for me, the correct way to do it is you get the test statistics, the 10,000 ones, okay, from the bootstrap, you order them, and you confront your observed test statistics to this order and put which quantile it will show up. So if it pops up on the extreme of the vector or any extremes, it will be, and then you get that basically the p-value will be the position if it gets in the, that's the p-value. You get a p-value for the bootstrap, not a 10,000 p-values and average them out. You see what I'm saying?
Yeah, I do. And what I mean, we thought about that, and the concern was that all the bootstraps are based upon one sample size that's fixed, whereas the empirical value is based on totally different sample sizes.
The empirical values has different sample size?
Yeah.
Oh, because of the 200 and 100?
Yeah. And that's those examples that we had. Yeah. But we thought about that as the point, and we thought about even comparing the p-value, empirical p-value to the adjusted ones.
Yeah, we need to think further about that, because I'm not convinced that you can take the average of p-values and so on and conclude on those.
So, no, we in fact don't think that it's this that it's just.
But I see your point. We need to come up with some correct. That I see on the test statistics, it's influenced by the total sample size, right?
Right.
Size. So then that's what you need to control for. Okay. That's it. So anyone, if you have any suggestions about that, that will be interesting to hear. Second point is when you get the minimum and the average, right, over that slide at the end, you use the minimum size, the average size. Clearly, if you get substantial evidence in the minimum size, you're making your life harder. So that's in the safe side. So that's how you play safe. If you get minimum and see evidence, you're good. The problem is what happens if in the minimum you don't see and in the average you see what's going on there, right? So since you have that control example that I'm showing right now, where you have basically, book kind of book data that you control for everything. So what I would do is I would get my marginal size to become smaller and smaller until I start to detect problems. So the conclusion from average becomes different from minimum. This way you can control empirically like a power curve,
Mm-hmm.
and it will help you. So if your, say, minimum size is above 100, it seems that you have enough power to use the minimum. So the power can help you to, so how to decide. If you have a minimum size of 3, but for the other months it will be like 50, and by chance 1 month was only 3 sketches, then you don't want to use 3 for all months because you're going to just destroy your thing. So, because, and you will know if I use 3, the power will be basically 0 to distinguish things. But if I go to the average, will be, I don't know, 45, I'll have enough power to make my decision. And I'm not sure that you will have exactly one way to do it, but at least with this power curve that you can always construct from your simulation, you should have a guidance to the authors. They will look at their minimum size, and you play with the marginal total, say from 20 to 100. You probably get enough. You will be able to construct this power curve. Your test improves as the marginal totals increases, right? And then you probably have this power curve to guide the readers to, okay, I look at my data, minimum, it's uniform, but the mean is around 25, then probably not enough power. They will at least have an idea what to do. This is what I take from your questions at the end. So these slides I'm showing there, if you want to go to the questions, maybe someone else can ask a question or so. But for the p-value, right, so that we need to think a little bit further, because for me, it's not correct to take mean of p-values or median or anything like that. But I agree, because you change the marginals, so then the test statistics will be dependent on that, and it's not fair to compare it to Bootstrap as well.
In
So
some senses, though, Mark, is the empirical p-values as well as is an adjusted test statistic based on sample size or degrees of freedom. So in some senses, comparing the p-values might be more informative than comparing the actual test statistics themselves. But I still think there's a problem there.
Well, the problem is, so, okay, it could be, but I don't think it's fair to say, oh, less than 0.05. That's the thing. It can give you evidence, but it cannot be used as a p-value. That's my point.
Exactly.
So it's not like, oh, this is significant because it's less than 0.01. I'm not sure what that means in terms of the probability space that it's looking at. But it's clear that it's some evidence, right? Because all the p-values are small, then, but I'm not sure how to translate that in a traditional fashion where people will select, say, a type A01 of alpha and check with that. I'm not sure how that will go.
Yeah, no, we concur. That's a big issue, both in terms of, well, that's a big issue.
So what happens if you fix the marginal size to be the same as you observe in the data? So each
you simulate.
Yeah, based on the proportion you have on the original data, you simulate new data, but fixing the marginals to be exactly the same as the ones you observe. What happened in that case?
We haven't done that, but we could. It would probably,
because that will respect the marginal totals, that will respect the sample size totals.
Yeah.
And the proportion will show. So that what would differ is that your count number from the bootstrap, it will come from the observed proportion you have, right? So that's the thing you use. So you randomly get a sample, it's pregnant or non-pregnant, given the proportion from the observed set, not the actual number. So if you have 80, you maybe have 44 and 36. And if you have 200, like I'm looking at the red one or orange one, right? 200 will be maybe a 110 and 90. It will respect that proportion, but not exactly the numbers. And it might be able to, and then you can order the histogram and get the p-value that way.
Right. So that's interesting. I know when sample sizes get small, we've had the test statistics themselves have some distributional issues when sample sizes are small.
Yeah.
And our initial approach was a pure Monte Carlo approach to trying to solve the problem, but it was it wasn't appropriate in the case of the second mismatch. We couldn't use it. It returned the wrong result for the mismatch that's in blue, in part because the distribution of statistics is so constrained at one end of that spatial distribution when sample sizes aren't the same. But in your case, see, what we were originally doing is we were pooling all the data to get the proportion of pregnant females. But in the example that you suggested, we'd still leave it as there'd be separate monthly
proportions,
pools that you would apply. So the problem may go away. So that's interesting.
I don't know. And the only thing I will know is you will carry all the problems. You have the sample size from the original data because they will preserve everything.
Right.
You make things comparable, I believe, in terms of p-values and so on. You can compare, but it may one of the things may break down. I don't know. We can try that. We can talk later and just run.
Yeah, no, that's cool.
Happens. Okay, so I assume from the audience here we don't have any questions right now. I didn't see any questions. So thank you for your time, and this will be saved on the YouTube, so you can revisit this and see my comments later or pass to your collaborators. They're probably watching anyways.
They are.
And if you think you want to talk again, and maybe I can get the accolades, try anything before to see if I can think on the problem. It's fine too. Okay.
Okay. Well, again, we thank you so much for your comments and for the opportunity to share this information. The three of us that obviously knew we don't have all the answers, and we're simply trying to. This talk was designed to kind of catalyze the kind of discussion that you and I've just had right now on YouTube. So we really appreciate your comments and those of your students.
It's nice because I assume at least 80% of our students don't know what a circular statistic was before this talk.
Right.
And it's very motivating to see this is not a theoretical problem. This is a practical problem that we need this. And there are many things to be improved because it's a difficult methodological problem as well. So if any of them are willing to work on this kind of situation, there's a lot of place for theoretical development, practical development, and so on.
Exactly. Well, thank you, Marcos. And hopefully the version of the talk that you have should hopefully all those if there's any infelicities, any slides where the data didn't transfer from my file to your file, let me know and I'll resend that to you.
No, mine, I went through it. It looks fine.
Oh, okay, cool.
Maybe when you upload it, if you upload another PDF, but the PPTX, that's maybe what caused it.
Yeah, I did. I uploaded the PowerPoint. Okay, but you have the PDF, so feel free to distribute that to your colleagues and students.
Sounds good.
Okay.
So, Mike, if you can stay on the studio a little, or maybe a minute, so
sure.
And I will just finish the transmission. So, thank you, everyone. So, next week it's also an online seminar from Adriana. She's presenting the Seminário 1B from her thesis, and hopefully we see all of you next week. Bye-bye.